في الدكتوراه أنا الطالب حسين جلاد، وهنبلش أول شي بإنه نشرح عن البروتين ستراكشر. طب هنا بأول سلايد حنوضح كيف إنه الأربع أنواع البروتين اللي عندنا. تمام، رح نحكي عنهم في السلايدات الجاية، وكل نوع بنحدد وظيفته والهيكل الهيكل ال 3D تاعه. البروتين ستراكشر عن البرايمري ستراكشر. هلا البرايمري ستراكشر أول شي بحكي إنه يشير التركيبة الأساسية للبروتينات إلى تسلسل أحماض أمينية، وبتكون بالعادة من 200 لألف حمض أميني، وبتربط ببتيد بوينت بين الكربوكسيل جروب والأمينو جروب. وبكون الباك بوند لجميع البروتينات بتحتوي على اليونت اللي هي بتكون N CAR وال CO. تمام، وبتختلف الآراء حسب تسلسل البروتينات، وبتكون مكتوبة من اليسار لليمين، يعني من ال N لل C terms. حنبلش في هذا السلايد عن secondary structure refers to short range periodic floating elements that are common in protein. These include the alpha helices, the beta sheet and beta terns. وكون البيتا strands then to occur in pairs and multi copies in beta sheet that interaction with one another via H bond directed perpendicular to the axis of each strand. Carbonyl oxygen and amino nitrogen in the strand from the H bond. Strand can oriental anti-parallel or parallel to one another in beta sheet. R group of every, of, of every other amino acid point, up or down relative to the sheet. Most beta strain in the protein are 5 to 8 amino acid long, and beta terns consist of 3-4. Mostly is stabilized by non-covalent interaction between secondary structure element and other internal sequence region. The folding of a protein is thought to be driven by the need to place the mostly hydrophobic region in the interior out of contact with water. Different methods of represent structure are shown in figure below, like X-ray and crystallography. راح نبلش بآخر نوع من البروتين ستراكشر هو كوارتر ستراكشر. والكوارتر ستراكشر له مستويات أخرى من تنظيم الهيكلي المعروف باسم الكوارتر ستراكشر، ويشير إلى number of subunit وزي ما هو موضح أمامنا في هذه الصورة ال A و B.
السلام عليكم، أنا بشر خانجي and I will talk about Alzheimer disease. Alzheimer is a neurodegenerative disease that slowly destroys brain cells and slowly destroys memory and thinking skills, and eventually the ability to carry out the simplest tasks. And there is 3 stages of Alzheimer disease. First stage, it's mild Alzheimer disease and also called early stage. The second stage is moderate Alzheimer disease, and last stage is severe Alzheimer disease. In this stage, in mild stage, a person may seem to be healthy but has more and more trouble making sense of the world around him. And there is symptoms in this stage like memory loss, taking longer time to complete normal daily tasks, and repeating questions. Alzheimer's disease is often diagnosed in this stage, in mild stage. About moderate or middle stage, more intensive supervision and care become necessary, which can be difficult for many families. There is some symptoms happen in this stage, in middle stage, increased memory loss and confusion, shortened attention span, repetitive statement or movement, inability to learn new things. In severe stage, people cannot communicate and are completely dependent on others for their care. Symptoms of severe inability to communicate, weight loss, loss of bladder and bowel control, skin infection, difficulty swallowing.
السلام عليكم أنا أمجد حسن راح أكمل بالstructure and function of tau protein. In pathological conditions such as the case in Alzheimer disease, abnormal phosphorylation of tau protein decreases its tubulin binding capacity leading to microtubule disorganization. Also, this protein self-polymerizes and aggregates in the form of neurofibrillary tangles. Tau protein belongs to a group of proteins called microtubule-associated proteins. It is famous for its heat-resistant and limited affected by acid treatment without loss of their function. This property observed in tau is due to a very low content of secondary structure, and many biophysical studies reveal that the tau is a prototypical natively unfolded protein. Since disordered proteins tend to be highly flexible and have variable conformations, they have not been amenable for structure analysis by crystallography so far. Thus, nuclear magnetic resonance spectroscopy is the only possible method that allows a description of their conformations and dynamics with high resolution. Six isoforms of tau protein differ according to the content of 3 or 4 tubulin binding domains of 31 or 32 amino acids in the C-terminal part of tau protein and 1, 2, or no inserts of 29 amino acids, each in the N-terminal portion of the molecule. These isoforms, which vary in size from 352 to 441 amino acid residues, are related to the presence or absence of sequences encoded by exons 2, 3, or 10. Inclusion of the imperfect repeat region encoding exon 10 leads to the expression of tau containing 4 microtubule binding repeats, while exclusion of exon 10 results in splicing products expressing tau with 3 microtubule binding repeats. Tau protein is rather a dipole with two domains of opposite charge, which can be modulated by post-translational modifications. Because each of these isoforms has specific physiological roles, they are differentially expressed during the development of the brain. For instance, only one tau isoform, characterized by 3R and no N-terminal inserts, is presented during fetal stages, while the isoforms with 1 or 2 N-terminal inserts and 3 or 4R are expressed during adulthood.
My name is Mohamed Abououn, and I'm going to start off with discussing the two main Alzheimer's disease biomarkers, which are the beta amyloid peptide and the tau protein. So, the two hallmark pathologies required for a diagnosis of Alzheimer's disease are the extracellular plaque deposits of the beta amyloid peptide and the flame-shaped neurofibrillary tangles of the microtubule binding protein tau. Tau proteins are a significant element of microtubule-associated proteins and are involved in the structure of microtubule network in neurons. Normally, the role of tau protein in the brain is polymerization and stabilization of microtubules. The hyperphosphorylation and accumulation of tau protein on chromosome 17 may be the sign of several neurodegenerative pathologies like Alzheimer's disease, Parkinsonism, and pig disease. So in Alzheimer's disease, the normal role of tau protein is ineffective to keep the cytoskeleton well organized in the action of the process because this protein loses its capacity to bind to microtubule. This abnormal behavior is promoted by conformational change and misfolding in the normal structure of tau that leads to atypical aggregation into fibrillary structure inside the neurons of demented individuals. Furthermore, most of the altered group of tau proteins in the disease are redistributed and aggregated in both the somatodendritic compartment and isolated process of the affected neurons. Alteration in the amount of the structure of tau protein can affect the stabilization of microtubules and the other processes related to this protein in Alzheimer's disease. Now speaking about the amyloid peptide Ys, which is the main cause of Alzheimer's disease. So the beta amyloid protein involved in Alzheimer comes in several different molecular forms that collect between neurons. It is formed from the breakdown of a larger protein called amyloid precursor protein. The amyloid hypothesis, which is the assumption that accumulation of the peptide amyloid beta, known as plaques, is the main cause of the condition. So we have glial cells that are normally meant to help keep the brain free of waste. To be more specific, the microglia engulf and destroy waste and toxins in a healthy brain. But when it comes to a patient suffering from Alzheimer's disease, the microglia fail to clear away waste, including the beta amyloid plaques. When amyloid beta clumps together to form deposits in the brain, it triggers neurodegenerative process that lead to the loss of memory and cognitive ability that is observed in Alzheimer's disease. Amyloid beta is therefore an obvious therapeutic target. So if you can deal with the peptide, then you can treat the condition.
I am Dia Kamal, and now I will start with the diagnosis and treatment for Alzheimer's disease. Until now, there is no accurate test that can determine if the person has Alzheimer's disease or not, but there are some tests to determine the presence of certain symptoms and ruling out other causes of dementia, like physical tests as see the reflex and the balance of the person, a mental test like see the ability to remember and do simple calculations, and there is a laboratory test like vitamin B12. Also, the brain imaging test as MRI, CT, and PET images can help the doctors to detect the brain change caused by Alzheimer's disease. Example of MRI image for healthy person brain on the left and an Alzheimer patient on the right. Other example for PET image of the difference between the healthy amyloid and tau protein that we talked about them in the previous slides and the Alzheimer ones in the brain. There is no cure for Alzheimer's disease, but there are some drugs that can help for a time with memory symptoms and other cognitive changes. Some of these drugs is cholinesterase inhibitors, Namenda, and aducanumab. The aducanumab drug is the first drug approved from the FDA to treat the underlying cause of Alzheimer's by removing amyloid plaques in the brain, but this drug is still under research.